For that, we prepared the excellent portal, make use of the Gotouniversity portal and Excel, attend all the classes, not only for physics, not only for chemistry, not only for maths, and even for biology, in all the grounds. We already had given, not only in terms of material, not only in terms of questions, also we given the relevant videos in addition to the worked examples. So welcome to Gotouniversity portal. Let me start discussing about the health and disease in detail. In yesterday's class, when we discussed about immunity, what are all the concepts of immunity we discussed? So again, back I'm repeating, what are the different concepts of immunity we discussed that will be more helpful for understanding the AIDS. So the AIDS is one of the subtopic of the health and disease, a continuation of the various type of immunity, right? So AIDS is not a immunity, AIDS is a disease. So why we need to discuss about AIDS in detail in this particular chapter? Why? Because when we are taking narcotics, when you are taking beverages, at the time we lose our consciousness, we are having the higher probability of doing the mistakes. For example, once you are losing your subconscious mind, unconscious mind, after taking the beverages, obviously you will be having the higher probability of doing the mistakes. Okay, in the sense to have a multiple sex partner, the maximum amount of energy production will be initiated because of the maximum ATP production. This is because of the maximum amount of ethanol consumption. So ethanol consumption predisposes you to get AIDS, number one. Number two, social media. For example, the visual reception and interpretation information. For example, when we are keeping on seeing the sex-related videos, okay, when you are keeping on studying the sex-related essays, okay, so what will happen is you'll be getting a lot of sex induction in your body, and that predisposes you to go for multiple sex. So when you are exposed to multiple sex, what will happen is you are having higher probability of getting the AIDS, number one. Number two, I already told, beverage intake. Number three, the narcotic intakes. Number four, the bad habit in terms of having the drugs. Okay, and then fifth important point is because of the misappropriate blood injection. For example, one person met with an accident. We are giving the blood transfusion to save the patient without doing the proper type of blood test. Then what will happen is the innocent injured person will get the AIDS because of the infected person who did the mistake. So these three number of probabilities we are going to discuss in this particular chapter in detail. AIDS, why AIDS is coming to one individual. For that, we already given so many number of examples, right? The expansion of the AIDS is acquired immunodeficiency syndrome. Acquired immunodeficiency syndrome is the expansion of the AIDS, which is caused by human immunovirus, which is a retrovirus. So we need to understand each and every concept in detail, in depth. Okay, one by one we'll discuss in detail. If we're talking about the AIDS, in the AIDS, basically the HIV spikes. If we're talking about the HIV spikes, which is very, very important for getting adherences or attachment to the target tissue. So what are the different type of target tissues you are observing in the case of human being body? Everything is not taken for consideration. Certain specific cell, all the microorganism, not like all the cells, it likes only a specific cell. For example, if you're talking about being HIV, I will attack macrophage, number one. So being a HIV, I'll attack macrophage. So number two is basically not only macrophage, okay? So helper T lymphocyte, I used to write like T to the lower script, helper T lymphocyte. So one is macrophage, I already told you. Other than this, what else are there? If you're talking about the modified monocyte is nothing but a macrophage. Why? Because in some board exam, they will say the macrocyte is infection site. Yes, obviously. This monocyte, once it is getting activated by the microbe, it will become macrophage, right? So this monocyte, after becoming activation, it becomes macrophage. This macrophage, helper T lymphocyte, and then neutrophil are the three infectious zone where the HIV will predominate. And then second thing is, this cells is a platform for the HIV. Okay, the modified monocyte or mature monocyte is called macrophage. helper lymphocyte and neutrophil, all these things are mainly responsible for making the HIV to get duplicated into many number. So this is happening because of many important reasons. See, listen, HIV is a communicable disease, of course. If you are in sexual contact, for example, let us consider if you're sharing the dresses or if you're sharing the water, if you're sharing the food, HIV infection will not come from infected to uninfected. So, when there is a illegal sex contact, when there is a multiple sex partners, when there is a improper drug intake in terms of injection, improper drug intake in terms of injection in the sense, for example, you know very well morphine injections, if you're consuming it, uh, definitely you are having a higher probability of getting the infection. For example, I am sharing the morphine injection from my friend. My friend did the mistake. He's having the HIV infection. So I not did any mistake. But when I'm having the injection from my friend, so what will happen is I'm having the higher probability of getting the HIV infection, right? So HIV is not a genetic disorder, right? HIV is not a genetic disorder, but it spreads from mother to the baby. Okay, it spreads from mother to the baby. So if the father is doing the mistake, mother will get the infection. If mother is doing the mistake, father will get the infection. If any one of the parent is doing the mistake, baby will get the infection. So this is not inherited. It is just transmitted from the body to the progeny with the help of the blood. So that particular transfer, I can't say genetic deficiency mediated transfer or genetic disorder transfer like this, we can't narrate, right? So we need to understand basically the AIDS is caused by HIV of various types. So the HIV belongs to retrovirus. Why? Because retrovirus indicates it's called back virus. So also called as, why it is called back virus, I'll say to you in detail with certain phenomena so that you'll understand. The retro in the Latin, it denotes the uh, black virus and then back virus. So retro. So retro, right? So now what we need to understand basically the HIV is having a RNA genome, even though it makes DNA. HIV is not having DNA every time. It is having the DNA for a while getting duplicated into many number. At the time, the HIV makes DNA for some time. It is not existing as a evergreen material in its body. Whose body? HIV body. Okay. This DNA is formed only for some time, not every time. HIV has the character controlling alleles in the RNA. In this regard, I'm calling the HIV RNA as a RNA genome. Why I'm calling HIV RNA as a RNA genome? Why? Because the RNA genome of HIV has the genes for controlling the various characters. The genes for controlling the various character is present in the HIV RNA. In this regard, I'm calling as HIV RNA as RNA genome. It makes DNA, of course, HIV makes DNA, but only when the HIV need to get multiplied. Okay, when the HIV is getting multiplied into many numbers. We all know very well the virus has both living and non-living property. So being a HIV virus, so outside the host, definitely I am not having the living property. So, but inside the host, I have the living property. For this, I already had given the example, not only the neutrophil, not only the macrophage, and even the helper T lymphocyte are all the target cells where the HIV get multiplied into many number, right? For example, when I'm coming to your birthday party, your father is inviting me with a hand, shaking my hand and making me to sit in one particular place, and then asking me to have a welcome drink. In the same way, the HIV, if it need to get adhered to the target cell, it need to have a receptor. It's also called as what? Welcoming structure. The welcoming structure or receptor of the specific white blood cells of human being body is giving a platform for HIV to get adhered, so that what? Furthermore, the HIV can target the host cells. Again, back I'm repeating, the host cells are nothing but white blood cells for the HIV. And this host cells are having the welcoming structure or receptor structure to which the HIV is getting attached. So for this to understand, we need to understand the HIV structure. So calmly listen to me. So can you see this place here? In this HIV structure, I am zooming and then I'm exposing this HIV structure for you to understand. This spike is made up of GP120. That means it is made up of 120 varieties of amino acids, right? So why we are saying GP120? Because it is having 120 amino acid in it. The HIV outer cover is called envelope. This envelope is very, very important for the HIV to get attached to the target cell. So you know very well the capsid is a cover for the DNA or RNA. Yes, of course, it is a cover for RNA. HIV has two single-stranded RNA, but not double-stranded RNA. HIV has two single-stranded RNA. Many of the students are doing the mistake by saying HIV has double-stranded RNA. No, it has two single-stranded RNA, which is the genetic material which is existing in the center of the HIV nucleus, right? So now what we need to know is the HIV structure is spherical in most of the books and syllabus. In the case of CBSE textbook, they already had given in this way, but HIV structure is not this. Okay, every time it keeps changing, but most of the time it is having the spherical shape structure. That is the reason why the scientist has given a spherical shape structure. The structure of the HIV is not consistent. Sometimes it goes to different type of bulged body structure. So like amoeba, like ugrina. So the overall outcome or conclusion of this entire information, what we conveyed for you is the HIV structure is keeping on changing because of the random mutation in the RNA. See, for example, let us consider my thoughts process, my insight, okay, the way how I am thinking very wrongly in my mind. In that way, I will work. In the same way, the HIV RNA, the way how it works, in the same way, what happens is the HIV structure also changes continuously. So why the HIV RNA is getting changes? Because of the ability to undergo mutation consistently. So the ability to undergo mutation consistently, why? Because this HIV RNA is having that many number of free hydroxyl group. So see, for example, let us consider, as I already told to you. So if I'm drunken, I am highly vulnerable for getting AIDS. So I will go to the prostitutes. I'll talk to the prostitutes. Okay, and then what I will do is I'll go with the AIDS, right? So in the same way, if we're talking about the RNA, it is having a lot of free hydroxyl group that is highly making the HIV to be more vulnerable, or else we can say another way around, susceptible or prone to get interaction with the mutagenic agents. So the ultimate single sentence, what we can say in a very concise way, in a precise way, the HIV RNA is highly vulnerable and getting mutated. Accordingly, the protein structure or the HIV is getting changed, and HIV structure is also getting changed. So though the HIV virus is getting changed a number of times in a microfraction of a second, so the being a doctor, we can't give a proper medicine for the HIV patients. And scientists are struggling a lot. We are not having medicine even now to kill the HIV, but we can stop the HIV multiplication. We can stop HIV multiplication, of course, but it is high side effects. But we can't kill HIV. So the God has not given the provision for us to kill the HIV. So that is the reason why it is more dreadful disease, because it conquers human being body at the maximum, right? So now, if you're talking about this entire concept of the HIV, being a retrovirus, it makes a DNA not every time in certain time period. In the sense, when it is getting duplicated into many number at the time it makes DNA, not every time, right? You know very well, as I already told to you, one is multiple sex partner, and second thing is improper transfusion of the blood. All these things will make the HIV to move from one person to other person. So, for example, if I'm sharing the food from with the AIDS patient, not a problem. Nothing will happen to me. HIV will not spread through food from HIV patient to me. Okay, for example, HIV infection will not spread through water. HIV infection will not spread through the food material. HIV will spread through only through sexual contacts, right? Now, if we're talking about the HIV of the particular human being body, after conquering the various set of target cells, what it does exactly, it creates a platform for its own to get duplicated into many number. Okay, so and also it switch off the different soldiers who are working against that. See, for example, let us consider So being a politician, what I will do is, for example, if I am a ruling party, what I will do, the opposition parties, corruptions, everything, I'll bring it to the maximum level in such a way that I will win in the forthcoming elections. So in the same way, the HIV also, what it does exactly, it exactly switch off the defense system of the concerned targeted human being body. If the defense system is completely switched off, then what will happen is all all the defense soldiers can't conquer the particular 8 patients. So again, back I'm repeating, the HIV is switching off the defense system of the HIV patient or AIDS patient. There is the first attack. Second attack is no defense soldiers to fight against HIV. Third attack is it takes all the cell consciousness to his consciousness, in the sense the HIV DNA, after getting incorporated into the target cell DNA, it makes the target cell DNA to uh, work according to its willingness. For example, being a hypnotic specialist, or taking the consciousness of the patient consciousness will work according to the hypnotic doctor. In the same way, the HIV DNA, after conquering the target cell DNA, the target cell will do the work of HIV, but it won't do the work on its own. So that is becoming a very good platform for the HIV to get duplicated into any number, right? So now if you're talking about the AIDS patient, um, or taken for various type of medications, okay, in the sense we can control the HIV, but we can't kill the HIV, as I already told to you. By means of this treatment, I can make the patient to survive for 10 years, but not more than that, right? So we know very well the HIV spreads from infected to uninfected person through body fluids that we already narrated for you. Body fluids in the sense not through sweat. Through sweat, HIV will not spread. Through saliva, HIV will not spread. So the sexual fluids, basically the external genitalia, in the case of male, we are calling as penis. In the case of female, we are calling as vagina. Not only those area, the area which are nearby to the penis, the area which are nearby to the vagina, also if it is in contact, without any proper sterilization, without any type of proper protection like condoms, then what will happen is the particular couple who are having the illegal sex definitely will get the HIV, right? So HIV was discovered for the first time in the world in the USA, basically in Florida. They found out this HIV patient for the first time in the world. So basically, this HIV is not only spreading from male to woman, male to female, or female to male. It also spreads from male to male when they're having homosex, and female to female whenever they're having the lesbian sex. Okay, like this, we are bringing all the varieties of HIV spreading probability to get the proper idea as per the CBSE textbook specification, right? So now we are coming for the next phase of explanation in the HIV. Basically, as I already told you, the macrophage is a landing platform for the HIV. So in the macrophage, sometime instead of using the word macrophage, multiple language are there. So what do you mean about multiple language? So you know very well the modified macrophage, like this we are not saying, the modified monocyte is called macrophage. So this modified monocyte, instead of saying like this, I can use a word as agranulocyte. So agranulocyte, the basic concept of the CBSE board exam preparation is multiple language usage to corrupt your mind in such a way we will put your mark down. In this mindset, the concerned paper setter will make the paper. So agranulocyte, example is monocyte. Monocyte is becoming macrophage. So instead of using macrophage, I can use monocyte. Instead of using monocyte, I can use agranulocyte. Instead of using agranulocyte, sometimes I can use WBC also. Okay. So macrophage is formed from monocyte. Monocyte, a type of agranulocyte. Agranulocyte, a type of white blood cells. So these all things are not there in the slide. Why we need to say this number of extra points? Why? Because in the board exam, they won't ask in a very easy way, discuss AIDS in detail. They won't ask how the HIV is infecting the cell. Like this, they won't ask. So they'll bring the confusion. So if they bring the confusion, you should be stable enough to face all the issues. For that only we are narrating this number of messages, right? This Gotouniversity platform is exclusively made for the students I'm not having money. Okay, my parents are not having that much financial credential to afford the this particular study. For them, this session will help them to boost their knowledge. With this knowledge, you can crack the CBSE with very good score. The other name of reverse transcript is not given in the slide. It's not given in the book. Okay, but they are having the connectivity with the other chapter, molecular base of inheritance. In this regard, instead of saying the reverse transcriptase, they can say the word called RNA dependent RNA polymerase. So RNA dependent RNA polymerase is the other term for reverse transcriptase. So, and then second thing, as I already told you, the other name of macrophage, in which way they will ask, I already told to you. So the macrophage is formed from the activated monocyte. Also, we can say differentiated monocyte. Either you say activated monocyte, either you say differentiated monocyte. Okay, is becoming a macrophage. All right. So if you're talking about the DNA integrase enzyme, how it works, I'll say to you one by one in detail. HIV makes DNA integrase enzyme, and that integrase enzyme, which makes the HIV DNA to get integrated into the host cell DNA. In the host cell DNA. To the host cell DNA, the HIV DNA is getting integrated because of the integrase enzyme. So the integrase enzyme is not there in this line. Okay. And the HIV DNA is getting incorporated. The host cell DNA is not there in this line. So the purpose of conveying this message is in multiple way, they'll bring the hidden sentence as a question and they'll make you to get confused in such a way you should not say the answer. So in each and every board paper, always they'll make 30 to 40% in this way, so that it should be a challenging. It should be challenging. It's not easy going, right? So I worked very hard throughout the year for many number of hours by sacrificing many number of my holidays. So the other student, okay, one of my friend Mohan, not studied anything properly. Okay, he will just prepare the last 10 years paper and he'll come and write the exam. He will get more mark because his handwriting is very good than me. That should not be a case, right? Okay, here the thing is basically we should evaluate the student based on the how much amount of effort they given for each and every subject, right? So that is the reason why they are asking the tough questions, right? So now, if we're talking about the HIV enters the helper T lymphocyte. So which lymphocyte? Basically helper T lymphocyte, right? So here, this helper T lymphocyte, basically its count is coming down. Why? Because HIV corrupted the DNA of the HIV corrupted the DNA of the helper T lymphocyte DNA. So HIV DNA corrupted the helper T lymphocyte DNA that I already written with the lower script. We can say subscript, okay, to the capital T, not a small h, with the capital H. So sometimes they'll use the word symbol. Okay, this symbol, if for example, if you're writing the capital T, capital H, that is wrong exactly. So most of the students are writing how we need to write the pH, how we need to write the helper T lymphocyte is a matter. Okay. So now if you're talking about the T lymphocyte count is getting down, the person is highly vulnerable for the infection. Highly vulnerable means what? Highly prone for getting infection. Okay, by the bacteria, virus, fungus, and parasite. Okay, HIV switch off the T lymphocyte, right? So what is exactly happening? So most of the microorganisms are trying to invade the particular HIV patient. When I'm keeping on saying this message, so if you are having the credential, the thirsty, knowledge thirsty should be there. Why many old age people died because of the coronavirus infection like this? Whenever you're keeping on thinking, the thymus gland size is very, very small than us. For example, okay, 60 plus old individual is having the small thymus gland than the 40 plus old individual. The 40 plus old individual is having the small thymus gland than the 20 years old individual, right? So the thymus gland, because of the age factor, size is keeping on coming down and not producing the sufficient quantity of T lymphocyte. So there is a reason why the old are highly vulnerable than the middle-aged people in terms of getting the coronavirus infection and capable of not resisting. Okay, capable of not resisting, we can say incapable to resist the coronavirus, right? Basically, in this particular area, the ultimate conclusion is T lymphocyte is coming down. Many microorganism takes over your body for its own survival, right? Okay, now how we'll do the qualitative analysis. So there is a lot of difference between qualitative analysis and quantitative analysis. Like this, we are saying the two important category. So QL and QN. So qualitative analysis we are doing, of course, with the ELISA technique, and the quantitative analysis we are doing with the ELISA technique. There are three different type of ELISAs are there. Of course, in the case of CBSE textbook, they are not told the three different type of ELISA. For the MBBS students, first year, we used to teach while doing the practical, the three type of ELISA. One is direct ELISA, another is indirect ELISA, another is called sandwich ELISA. So one is direct ELISA, as I already told you. Another is indirect ELISA, another is what? Sandwich ELISA. So DINS, which is not there in the CBSE textbook. That is why we are having the control over the tongue. We are not going outside of the NCERT textbook, right? So this different type of ELISA is used to do the qualitative and quantitative analysis. Qualitative means HIV is there or not. That's it, qualitative. Quantity, how much? Accordingly, I will be giving the medicine being a doctor. Okay, this medicine calculation also depends upon many other parameters like associated disease. For example, if the concerned patient is having high BP, if the concerned patient is having diabetic, if the concerned patient is having the nausea, runny nose, like this, so many other factors also will consider. Accordingly, will be given the medicine, right? So you know very well the HIV is coming under a important category, retrovirus. All the retrovirus are not curable like this. I already not told. You know very well influenza virus is a virus, but not a bacteria like Haemophilus influenza. Haemophilus influenza is a bacteria, but influenza virus is totally a virus. So that influenza virus, why you used when we are discussing HIV, in which way it is getting interconnected, why you are jumping like this, you should not think in this way. Why? Because the curable retrovirus is influenza virus, okay? Whereas non-curable retrovirus is HIV virus, right? So curable retro influenza virus, non-curable retro HIV. So like this, we need to think in many ways for getting the better clarity. Now, when we are entering into the next important antiviral drug or retroviral drug can stop only the HIV multiplication. We already know very well that syringes, whatever we are using for the HIV patient should be discarded. We should not use for other patient. If that is the case, then what will happen is the innocent other patient also will get the infection, even though they are not doing multiple sex illegal activity. Okay, now we are coming for the next important category is the how to prevent the AIDS. See, for example, here in the developed countries, and I'm saying about in the great well-renowned great scientists, you know, when they are working continuously for 12 hours to 14 hours, when they're keeping on working, definitely because the overstress, okay, they need to have this type of multiple sex activity in this particular area. Also, we are giving the instruction to the people to have a safe sex by wearing the condoms and also by wearing the important shield. In such a way, you won't get the HIV infection, right? So you see, one is either use male condom or either use female condom, so that what the particular partner will be not getting infection. For that, we are giving the advertisement. Okay, here the drunkards. Okay, here the drunkards are highly prone. Why? Because once they are taking the drinks, obviously their ATP level will go for maximum breakdown. As a result, what happens is the max amount of energy will not make you to keep calm. So obviously you do mistake, right? So obviously you will try for, okay, I need a certain more relaxation, so I need to do the sex relation. Like this is what your brain will predisposes you to have a multiple sex partner. So because of this, you are getting the HIV infection. That is one thing. And second thing is social media. That is one thing. When you're keeping on seeing the sex movies and videos, so your sex stimulus, sex stimulus area of the brain. Sex stimulus area of the brain. Okay, how do we call that as amygdala? So amygdala is very, very sensitive area in your brain. For example, you know very well, I'm showing in my brain, This is the forebrain, midbrain, and hindbrain. The last segment is called hindbrain. In the hindbrain, amygdala is present? No. In the forebrain, amygdala is present. The amygdala is little sensitive. So little sensitive. Visual reception and interpretation. Hypothalamus is also doing this work. So this visual reception and interpretation, while keeping on seeing the maximum number of sex movies, obviously you can't control behind certain limit. So you are predisposed to have a sex, right? So now we are coming for the next major segment. Before discussing about the cancer in detail, let me start discussing about a few questions. What are the different type of questions will be asked? How does what does the AIDS stands for? As I already told you, acquired immunity deficiency syndrome. So there is a chance for asking differentiate syndrome and symptom. Symptom, for example, I'm coughing, I'm sneezing, I'm shivering, I'm shaking. So these are all the symptoms. The syndrome is nothing but a group of symptoms, right? So the answer is acquired immunodeficiency syndrome is the correct answer for the AIDS. So now we are coming for the question number 2 is, see, they will ask a lot of technical questions. How is HIV primarily transmitted? Through body fluids, through physical contact, of course, through the body fluids. Body fluids means not saliva, not saliva. Okay, semen, vaginal fluids, rectum fluids, breast, in all these things, the HIV spreads very faster, right? So now we are coming for the next major question is, which of the following is not a common mode of HIV transmission? You know very well, the HIV is transmitted sexual contact with the infected person. That is a correct option. So all looks very similar. Okay, sexual contact, sharing needles, and from mother and child, right? So which is the higher probability? Again, back they will give the higher probability, right? It's not a common mode of HIV transmission, right? Mosquito bite. So mosquito bite. Through mosquito bite, it is not getting transmitted. So listen, because of the exam tension, okay, and because of the failure in pre-calculation for each and every minute, how I need to spend for writing each and every answer at the time, if you're not calculating appropriately, what will happen is we predisposes or we pushes our body to do the answer very faster. As a result, what will happen is while doing very fast, you won't read the sentence properly. Which of the following is not a common mode of HIV infection? Not, we will not see. So we'll choose the wrong option. So be very careful. Not. Mosquito bite is not responsible for HIV transmission. Through mosquito, to none of the human, the HIV is transmitted, right? It is a vectorless disease, non-curable disease. Now we are going for what is the role of reverse transcriptase in HIV life cycle. If you're going to the HIV virus life cycle, calmly, if you're listening, we can make you to understand. As I already told you, the HIV RNA is making the HIV DNA. This HIV DNA is formed with the help of an enzyme called reverse transcriptase enzyme. So we know very well the reverse transcriptase enzyme is an enzyme, okay, which is responsible for making the HIV DNA. So this reverse transcriptase is an enzyme, okay, which makes the HIV DNA. So what else we should suppose to know is the HIV DNA is mainly responsible, HIV RNA is responsible for making the HIV DNA by using the reverse transcriptase enzyme. So this HIV DNA is getting incorporated into the host DNA and making the host DNA to make the HIV components, but not the host components. The host components are completely coming under the control of the HIV DNA, right? This is a message we need to understand. Again, back, I'll repeat for you. You know very well the HIV RNA is making the HIV RNA makes HIV DNA, and HIV DNA is getting incorporated into the host DNA. And in the host DNA, what happens is exactly the host DNA, after getting the incorporation of the HIV DNA, it does the HIV component synthesis instead of making the host component synthesis. So this is the way the host is ruled by the HIV RNA, right? So this is one part and this is the other part. So keep the concept in the mind very nicely, and then we'll go to the next concept. Before entering the cancer, furthermore, we need to do not only with the questions. These are all the questions are taken from the NCERT textbook, NCERT exemplar, hard questions of the CBSE board for better practice. The relevant video, whenever you're having time, please go through the video, which is loaded in the Gotouniversity portal. Examples, worked example. Can you see this? Always in the worked examples, describe the mode of transmission of HIV and how it leads to AIDS. Can you see this? So as we already told you in the worked examples, n number of subtopics, and again back under each and every subtopic, many bullet points. If it is narrated with the proper underlining, you secures much more point in terms of marks. We know very well in this particular question, the occupational exposure. Okay, healthcare workers may be at the risk if they're accidentally exposed to the HIV infection. For example, here I'm a surgeon assistant. So while doing the surgery, very terrific surgery, so I had 2-3 cuts in my skin. So what will happen is I'm going to the next treatment. I'm lifting the HIV patient who is having the lot of blood leakage. So that blood, when it is exposed to my body, okay, even though I'm not having a sexual contact, so there is a maximum probability of getting the infection. So the blood contact, okay, and then the rectum fluid contact, semen fluid contact should not be there between the healthcare workers and between the patient. If that is the case, then what will happen is we are having the higher probability of getting the infection. So always in the case of board preparation, for example, your answer bundles will come to one particular school. So there in that particular camp, when we are evaluating, so we will see how many subheadings you already had given, and then how many keywords you covered, and how long you explained all the concept in a very precise way. In a very precise way, because everybody are not having time to read the paragraphs in depth. So we need to bring the technical concept. See, for example, let us consider if you are keeping on writing the social science concept. Okay, your AIDS patient should be very careful. They should not drink anything like this. If you're writing social science concept, what they will do, they will not give marks for you. So we need the technical base, CD4. What do you mean about CD4? Complementary determinant. 4 receptors of the T lymphocyte makes the HIV to get hangover. The HIV is getting adhered or attached to the T lymphocyte because of the CD4 receptor. So these are all the technical sentence, right? So those technical sentence, how many you load in your answer sheet? After loading, what all the things, how you underlined it, and how you are representing to us, not only in terms of subparagraphs, not only in terms of points, and also how you are underlining the important keywords. All these things will make you to secure much more mark. Here, now you need to understand it is not only spreading from mother to child, it is not only spreading through the needles, not only through the blood transfusion, and also occupational disorder, occupational exposure, like this we told you, right? That also we need to narrate. Now we are entering into the other question. Explain the importance of antiretroviral therapy in the management of HIV. So in the antiretroviral therapy, the medicine name they are not already had they are not given in your CBSE textbook. So we are having the control over our tongue. But in many of the state board book of Andhra, Maharashtra, and all, they given the medicine name. That is nothing but here. Any one of you can say the answer for this? Okay. Crixabidin. Crixabidin is a medicine. Is given to the HIV patient. And that medicine name and all, they're not given in the book. That is why what we are not writing much more and explaining much more about the drugs. So the HIV patients, after appropriate screening, in the sense, after understanding the amount of HIV in the body, I can give the dose accordingly. But we can't kill the HIV, as I already told to you. We need to understand the virostatic. See, virostatic is not there in the slide. That is why what I'm searching for the keyword. Okay, why? Because in the board exam they will ask static is there or not, cidal is there or not. So one is static, another is called cidal. Cidal means killing property. None of the medicine is having the HIV killing property. Okay, again back I'm repeating, cidal property is 0. Cidal property is 0. Static property is there much more. So what do you mean about static property? So static means I can stop the HIV multiplication. That drugs are available in the market, but cidal property drugs or the drugs which are bearing cytal property for killing the HIV is not discovered as of now. Like this, what that also we need to narrate. Next major segment is, if we're talking about the HIV of various structures and shape, we already discussed with its RNA information just now only before 20 minutes back. We know very well the HIV is having the efficiency to spread not only in blood body fluid, not only through semen, not only through vaginal fluid, not only through the anus fluid, not only through the rectum fluid. So here we need to think in multiple way. What all the fluids means? Just randomly, if you're saying body fluids means that also will not give the marks. What body fluid exactly? Saliva, through saliva, HIV will not spread. Through running nose, nausea, HIV will not spread. So through which body fluid HIV is spread? Through which body fluid HIV will not spread? That also we need to narrate if they're asking such a type of question. Right now, we need to understand the HIV spreading mode. HIV spreading mode that we already discussed in detail. But after getting entry into the body, HIV stays for some time in an inactive form for a while up to its invading point. See, you need to understand basically the HIV. If you are talking about the latent stage, what do you mean about latent stage? Means inactive stage. So also called as what, prophage. So either you say prophage, or either you say latent stage, or either you say inactive stage. So in this stage, what happens is HIV will remain inactive for a while in neutrophil. So it is not in a position to infect any of the target cell. So it takes some time. Neutrophil, the spelling, whatever you written like this is a die. Many students, because of the hurry-burry, because of the exam tension, they are writing neutrophil. Okay, E should not be there. E should not be there. Okay, E neutrophil is nothing but a bacteria. Okay, in one contest. In other contest, neutrophil is a dye. So we need to say neutrophil, Neutrophill, without E. This neutrophil is a platform for the HIV to get developed from the inactive stage or latent stage or prophage. Like this, we need to narrate, right? So be very careful with all these things, okay, in which way they're asking the question. Accordingly, you need to fine tune your answer script. If you're talking about in this place, the related video examples we discussed, tips for exam, we'll go to this. In this particular junction, I need to understand all the terms, HIV, CD4, antiviral therapy. So the term checking is very important. I need to underline the important words, okay, for better correction. And second thing is focus on prevention, how the HIV is prevented. And the second thing is, if you're talking about the importance of HIV testing is very, very important. See, for example, we all gone for get together in hill station. So we took drinks and we had sex, but I still have the fear, may or may not. Okay, I am feeling that might be HIV would have entered into my body. So if there is a case, so please check your body, go for HIV test. So do not worry about it. Either it is positive or negative. So if it is positive, we'll give the treatment. Okay, so that your survival span will be increased. So if you are keeping on doubting and not going for testing, what will happen is we can't give the medicine and your lifespan is reduced. So don't be panic. If you would have done the mistake and have the proper diagnosis and then treatment. So this is the way the diagnosis and treatment narration should be given in your answer script.